Good afternoon. My name is Chris Suarez. I'm the Scientific Applications Manager for Corning Life Sciences. Thank you so much for joining us this afternoon. I'm really excited to talk to you about the 3D cell culture techniques that we have available for modeling development disease within the Corning portfolio of products. There are several key areas I want to focus on during our time together this afternoon. The first will address the question of why 3D cell culture. We'll talk about the difference between 2D and 3D cell culture briefly, define the difference between spheroids and organoids, talk about the trends in research applications moving towards 3D. We'll then transition to a focus on extracellular matrices, or a scaffolded approach to 3D cell culture. This will include introduction of Matrigel, collagen, and even use case scenarios for both Matrigel and collagen. We'll then move into both a scaffold and scaffold-free based discussion. This is where some of these tools and techniques will intermingle. Things such as the spheroid microplate, our Alplasia microplates, even Matrigel Matrix 3D plates, and also our new product, the Matribot, which will help to bring some automation to the 3D cell culture workflow, utilizing a scaffold-based approach. I'll then move into 3D applications that we've worked on at Corning, and those involve airway organoids, intestinal organoids, and a model of a blood-brain barrier, a glioma-based application. Some of the things that we want to be aware of when we're looking to move from a 2D to a 3D cell culture model are the ease of use, the biological complexity, and the throughput capabilities of the model that we would choose. In 2D, we have very high ease of use. We've got very high throughput. What's lacking is the biological complexity. Now, of course, the mouse model gives us the highest biological complexity, and our 3D cell culture models, being the spheroids and organoids, fall right in the middle, right? So they still maintain that ease of use. They give us better biological complexity and also are amenable for high throughput application. Two-dimensional cell culture models are frequently used in drug discovery due to their ease of use and established compatibility with high throughput screening. However, as we talked about on the previous slide, we need to capitalize on having models that more accurately recapitulate development and disease states for tissues. Now, the results from 2D-based cell screens don't always translate into success in clinical trials. Furthermore, the culture of cells in 2D adherent monolayers, they do not accurately represent the environment that cells experience in vivo. There are more elaborate three-dimensional cell culture models that have been developed and offer significant improvements over traditional 2D monolayer cell culture in terms of maintaining morphological and functional characteristics of tissue. For example, 3D cell culture models more closely mimic the in vivo tumor environment, as 3D cell cultures develop hypoxic cores and demonstrate gradients of various soluble factors and a diffusion profile for drugs similar to tumors. 3D cell culture models are increasingly being incorporated in drug discovery as model systems, again to study development and disease states, and for screening of chemotherapeutic efficacy or drug toxicity. I introduced the terms spheroid and organoid on a previous slide, but let's take a little deeper look at what defines the differences between these two 3D cell culture models. A spheroid is a more simple spherical cell culture model, typically without organ-specific characteristics, whereas an organoid is a more complex and organ-specific 3D cell culture model. There are several different areas where there have been advancements in 3D organoid applications. Those include areas such as regenerative medicine, ADME-tox or drug safety testing, as well as development in disease modeling that can then filter into applications for drug screening and drug efficacy testing. Now again, this closes that loop into a more personalized medicine approach where oftentimes you can look at taking a diseased and healthy tissue from a patient biopsy sample, use that in a 3D cell culture model that then goes forward into drug efficacy testing that's specific for that patient. Just like we talked about various application areas for 3D cell culture, there are also various ways to go about forming organoids for your particular assay. Those include both scaffold and scaffold scaffold-free approaches. The scaffold or gel-based approach can involve things such as Corning Matrigel Matrix, our collagen, or our more recent development in this space is our Corning Matrigel Matrix 3D plates that have Matrigel already aliquoted into the wells of that microplate. There's also several products that you can generate organoids using a scaffold-free approach. With our typical flat bottom, ultra-low attachment surface products, you can generate various size organoids. So these are multiple non-uniform organoids. You can also use that same ultra-low attachment surface in our spheroid microplate to generate a single uniform spheroid per every well of that microplate. Using the Alplasia plates, you can generate multiple uniform spheroids in these several cavities in each well of the microplate. So this gives you scale-up capabilities if you need to bulk produce large quantities of spheroids or organoids. Now that we've talked about the various ways that you can go about choosing to form spheroids or organoids for your assay, there's also the next question of which method should I choose for my particular assay. So I've got three different images here, one of an embedded culture, the middle here is a sandwich culture, and the last being a dome assay for spheroids or organoids. Now, when trying to make the decision on which method to choose, this can be variable and oftentimes, can be dependent on whether you plan to do imaging or you're just growing these for harvest and downstream analysis. So in that case, for embedded, the reason we put this image up here is you can see that in the background you have a lot of out of focus organoids that will interfere with having a cleaner image. Therefore, when you put these spheroids or organoids into the ECM, again, you're going to have different X, Y, and Z positions. That makes it very difficult to control for the out of focus interference from the background organoids. Now in the sandwich culture, you can make that a much easier transition by first laying down your ECM, right, your Matrigel or your collagen layer, polymerizing, and then putting your spheroid or organoid starter cultures on top of that. This gives you a much cleaner focal plane, right? So now you're controlling for a more uniform Z position when you're doing imaging applications. And then with the dome-based assay, you also are controlling for not only Z being more confined with these dots or these domes, you're also controlling for that X, Y, and more uniform Z position by putting the different domes, multiple domes per well in this plate. So you can see a representative image of that dome assay and the spheroids or organoids that are in that ECM. Now in this previous scenario, when I was talking about how to pick the right imaging assay, you'll notice that I was focused on the scaffold approach, which requires extracellular matrices. So let's quickly talk about the importance of extracellular matrices when doing 3D cell culture. So ECM proteins are secreted by cells into the extracellular environment of tissues and organs. Now the roles of the ECM are critical for 3D cell culture because they involve so many different functions, right? So they impact growth and proliferation, motility, morphology, differentiation, receptor-mediated signaling, and gene expression, right? So there's a lot of impact from the ECM, right? There's a lot of different things to consider. Those include cell-to-cell and cell-to-ECM interactions, right? The stiffness of that ECM, the things we talked about previously, the oxygen, the nutrient, and metabolic waste gradients, and also the tissue-specific scaffolding cells that are involved in that paracrine signaling. Building on the importance of the role of the ECM in your 3D cell culture, we got this example from the literature showing the impact of prostate organoids when they're cultured in collagen versus a Corning Matrigel matrix. Here, just looking at the morphology first when they're cultured in collagen, you can see that these structures are very elongated, very separate, right? Not a uniform, consistent spherical shape, as is seen when they're cultured in the Matrigel. Now in the Matrigel, you can see a very tight, compact sphere structure in each of these three different cell lines. Again, illustrating the importance of the role of the ECM and the particular choice of your ECM when you're performing your assay. In a different example from the literature, I wanted to highlight the impact of ECM choice on mammary organoids. Now, in this particular example, the choice of, say, a Matrigel without growth factor allows completion of a cyst formation assay. And this image on the upper right-hand side shows you the formation of that cyst over time, again, using Matrigel without the growth factor. Now, if you look at sets of images in panels B and C and even D, these are examples of a branching morphogenesis assay. Now, this can be accomplished with a choice of Matrigel with growth factor, or even a combination of Matrigel and collagen containing growth factor. Now, in the last example here, an invasion assay can be completed, again, when you go back to using just a pure collagen ECM, where you see this morphology of protrusive tips using just a collagen compared to a combination of Matrigel and collagen that results in this more rounded end on this particular organoid. And so here again, a good example of sometimes the combination of ECMs will result in a different endpoint assay, and this is important during your considerations of setting up your 3D cell culture conditions. I've talked about the Corning Matrigel matrix in these ECM or scaffold-based approaches, and it continues to be one of the gold standards when doing 3D cell culture. We've got listed here the components in the regular Matrigel matrix. It's composed of laminins, collagens, intactins, MMPs, and different growth factors. This is a natural hydrogel. Right? It's extracted from EHS mouse tumors with the goal of this mimicking the ECM composition and structure that is found in vivo. Now, in addition to Matrigel, right, and I've talked about even in the previous slides with the mammary organoids, the different formulations that are involved in different assay endpoints. So we have standard formulations, growth factor reduced, phenol red free, GFR and phenol red free, high concentration and stem cell qualified versions. Now what's nice on this slide is I'm also showing the more complete portfolio that Corning offers for complex and more defined cell culture attachment options. Again, if you look at the image here, the biological noise moving from left to right is more defined, right? So you're moving to more defined attachment, which are sometimes singular components, whether that's collagen or laminin. If you move a step to the right, the design xeno-free attachments, right? Again, we've got human collagens, fibronectin, osteopontin, vitronectin, and then also defined synthetic and animal-free. So things such as our peptide hydrogel Puramatrix, our Synthemax, and our PDL. We'll shift gears here to a scaffold-free approach and talk about the technologies for 3D cell culture, such as our spheroid microplate, that allows 3D spheroids to be grown in suspension. Before describing the ultra-low attachment surface and how it's able to keep cells in suspension for a 3D cell culture application, I wanted to briefly review how cells interact with the tissue culture treated surface in 2D. I'm highlighting here these little blue ovals that are representative of, say, fetal bovine serum protein, which binds to the tissue culture treated negatively charged surface. This will be the basis and the foundation for cells as they settle and interact to form integrin cam-based interactions that will allow that cell to flatten out into a monolayer in this 2D attachment to a TC treated surface. In contrast to what we saw with the tissue culture treated surface, the ultra-low attachment surface is not negatively charged, but instead is a neutrally charged hydrogel surface. With that neutral charge, we don't have the same propensity of these attachment proteins to be on the surface and attached to the surface. Therefore, when the cells go to settle down to the bottom of this plate, they won't bind and be forced to flatten out into a monolayer, but instead they'll be able to form the initial structure by binding to each other of a 3D spheroid or organoid. I have highlighted here the features specific to Corning's ultra-low attachment surface. Now this surface is designed for maintaining cells in a suspended or unattached state, as we've discussed. It is a covalently bound hydrogel coating. It's hydrophilic and neutrally charged, and that results in a hydrogel that inhibits nonspecific immobilization and protein binding. It is stable, non-cytotoxic, and non-degradable. This image on the right-hand side is showing a tissue culture surface with the glioma cell colonies, again, forcing them to flatten out into a monolayer. Whereas on the ULA surface, we see the formation of these glioma cell colonies as 3D structures. And this is because, again, they're not being forced to flatten out into that 2D monolayer. The properties of that ultra-low attachment surface were utilized when we developed the spheroid microplate. This allowed us to generate a plug-and-play design that allows for growth in the assay being performed in the same plate. Through this workflow, you go from plating cells in step 1 into the spheroid microplate. Using the ULA surface, those cells will settle to the bottom of the conical round bottom on that plate, forming a spheroid, and then you're allowed to, in step 3, move directly into your assay without the need to transfer your spheroids to a different plate. This takes advantage of the black sidewalls for luminescence, fluorescence, colorimetric readout that in stage 4 allows you to feed this directly into your microplate or your high content imager to generate your data. All right, when starting to work with the spheroid microplate, it's important to look at optimization. That can involve looking at optimizing the seeding density and also checking things like viability over time, because these will combine to aid reproducibility when you go to repeat these assays. Here on the top set of images, you're seeing 4 different cancer cell lines at varying seeding densities from 40 cells per well up to 10,000 cells per well. Now the thing to consider, right, the different size spheroids or organoids that you're starting with and how long you intend to culture those organoids can have an impact on how the viability, right, will be impacted over time. So of course, if you start at this 10,000 cells per well, it's likely you could experience a necrotic core, right? Some of those cells losing viability if you intend to culture that over a longer period of time. This is why we have on the lower panel just an example of the HT29 cells that were cultured for 3 days. An example timeline of what we're looking at with the viability readout here beyond 48 hours. This can impact as you start to see some of that fluorescence dimming in the core of that spheroid. This can be an indicator that you're losing viability of those cells. So these 2 studies, again, are important when you're starting to work with the spheroid microplate to understand what is the optimal seeding density and what is the intended timeline and potential impact on things like viability when you're starting to lock in some of those parameters. In addition to our spheroid microplate, we also have tools to help with scaling up or bulk production of your spheroids. These are the Corning Alplasia microplates. We do have options that have an ultra-low surface attachment, so you'll have consistency between the spheroid and the Alplasia. We also have options that will allow you to do self-coating in the Alplasia microplates, with the end goal being, again, bulk production or scaling up of your 3D cell culture. More specifically, our round bottom Alplasia plates have the ULA surface. These have 500 micron well diameter. Here you can see some of the numbers on the round bottom. In a 6 well, for example, you get just under 3,000 spheroids per every well. This gives you an idea of the ability to scale. And as I mentioned, the contrast here is something like the spheroid microplate. You're getting a single spheroid per every well, whereas here you're getting those multiple spheroids in the Alplasia plates. Now compared with the round bottom, we also have options in the square bottom. Now the difference is our square bottom plates are plasma treated for self-coating, and these have 200 micron diameter per well, which as you can see in the table on the right, results in overall much larger bulk scaling. So for example, now in the 6 well, you can get just under 16,000 spheroids per every well. Now the good news, regardless of whether you choose the spheroid microplate or the Alplasia microplate, that same plug-and-play approach and workflow can be maintained across both of these plates. Right? So general workflows involving either the spheroid or the Alplasia microplates, you go from that plating, right, moving straight into generating your spheroids as those cells just settle to the bottom of these wells. You can form the intestinal organoid in this particular example. If you need to even combine, right, so we'll get into this a little more in the applications, but in this scenario, you can go into a scaffold-free directly into a scaffolded approach where you can then add Matrigel to the spheroid microplate to generate a more complex organoid. In this particular example, we use a 96-well spheroid microplate to generate several intestinal organoids. I'll talk more about that in the later slides. And that same workflow is maintained when you're looking at the Alplasia microplates. So from plating to allowing these cells to settle, generate multiple spheroids per well. Again, similar design with the black sidewalls. You can move directly into luminescence, colorimetric, or fluorescence-based assays. Got some examples of what those images look like in the bottom panel here in the middle. And ultimately, if that's not your endpoint readout, you can also use Alplasia, as I mentioned before, for bulk production. So if you needed to easily harvest those spheroids for downstream applications, keep them in a dynamic culture condition to continue expansion or maintenance, you can do that as well with the Alplasia plates. Building on this idea of scaling up with the dynamic culture, I wanted to quickly just introduce some work that's been reported that combines both static and dynamic culture conditions for 3D cell culture. In reviewing this work from 2019 out of the Arlata lab at Harvard, there were several key principles that I wanted to share that illustrate this concept of going from a static condition into more dynamic conditions for modeling development. Going from the initial 96-well microplate, you can see that they were able to go into another ULA compatible dish, but at this point they're using 70 RPMs on an orbital shaker inside of an incubator. This helps to control for the size before ultimately moving into a spinner flask that again will allow further development and differentiation of those brain organoids. You can see on the images on the right, regardless of whether it was whole brain organoids, it was the dorsal forebrain organoids, the patterned dorsal spheroids, or even the patterned ventral spheroids, that these conditions going from static into dynamic spinner flask did allow for reproducibility for these 4 distinct protocols to produce 3D brain organoids. As discussed on the previous slide, one of the key factors of going from a static to an initial dynamic culture condition is bulk scaling capabilities with the ULA microplates. Now our Alplasia microplate consistently will allow that ULA surface to generate multiple spheroids per every well. And again, we'll give a different approach to generate uniformly sized spheroids before going into dynamic culture conditions such as the spinner flask. Now, just as I've talked about this ease of use and high throughput application for our plug-and-play spheroid and Alplasia plates when you're using a scaffold-free approach, we wanted to have that same ease of use and high throughput application, and this is why we developed our new product focused on Matrigel Matrix assay-ready 3D plates. The Corning Matrigel Matrix 3D plates are offered in both 96 and 384 well formats. These plates are easy to use and provide the convenience of a pre-distributed Matrigel in high throughput formats, while providing the consistency required for scaffold-based 3D assays. The use of Matrigel has been demonstrated to enable polarized epithelial structures to form in vitro and can be used to create a wide variety of 3D cell culture models. The 2 examples of applications that we have worked on at Corning involve using MDCK cells to study polycystic kidney disease and generating an airway organoid model for virus infectivity research, which I'll discuss in more detail on the following slides. To generate the airway organoids, we plated human bronchial epithelial cells on a 96-well Matrigel matrix plate. Within 3 days, we saw formation of the airway organoid structure, and after 28 days, those airway organoids had grown to approximately 150 micron in size. And as seen in the lower set of images, this was consistent in size between individual wells of that 96-well 3D matrix plate. We confirmed that the airway organoids contain multiple cell types. The presence of proximal airway epithelial cells in the organoids was confirmed with positive staining for beta-4 tubulin, seen here in the middle image on the top panel. This is indicative of ciliated cells located on the apical surface facing inward towards the air-filled lumen. In addition, the presence of MUC5AC confirmed goblet cell presence, as well as basal cells positive by the cytokeratin 5 staining. Immunofluorescence staining was performed to investigate the presence of virus-specific host cell entry receptors in the airway organoids. Experiments confirmed the presence of ACE2, the receptor for the SARS-CoV-2 virus. Higher ACE2 expression in airway organoids relative to 2D cultured epithelial cells has been observed in other reports, supporting the utility of the airway organoid model for coronavirus research. Now, in addition to ACE2, we also saw presence of SNA. SNA is a lectin histochemical stain that binds preferentially to alpha-2,6 linked salic acids, which are the empty receptors of human influenza virus. The binding of SNA indicated expression of this alpha-2,6 linked salic acids, which is consistent with data reported in primary lung airway organoids and ex vivo cultures. Now, in addition, co-immunostaining of ACE2 receptor with the ciliated cell marker beta-4 tubulin and the basal cell marker cytokeratin 5 indicated that this higher expression of the virus-specific marker was present in basal cells. This data is comparable to previous literature reporting that basal cells were positive for ACE2 with expression at the outer edge of the human bronchial organoids. In addition to the airway organoid work that we just described on our new Matrigel Matrix 3D assay-ready plates, we realize that there's a need for continued automation and innovation in the 3D cell culture space. With that brings the opportunity for us to introduce the MatriBot bioprinter. The MatriBot bioprinter will help to facilitate and to bring in an automation component to your 3D cell culture workflow. The Corning MatriBot bioprinter is a cost-effective unit for researchers to enter the 3D cell culture and bioprinting space to start bioprinting living tissue models with ease. Now, the key features here are it has both dispense and print capabilities. That does include multi-layer printing capabilities. It also has temperature-controlled print head and print bed. It's in a compact size and has an intuitive programming interface. To speak more specifically about the MatriBot capabilities, I want to highlight some of those key features so that you have an idea of exactly what the MatriBot can offer. The small footprint does allow the benefit of taking the benchtop unit into a hood or a biosafety cabinet. The cooled syringe print head will allow you to use Matrigel Matrix or collagens or other room temperature hydrogels. The thermal nozzle insulator is going to reduce clogging at the nozzle, again for temperature sensitive hydrogel work, as well as a curing system that has a UV with a 405 nanometer LED and the temperature of that print bed, which is going to give you multiple modes to cross-link hydrogels and maintain your 3D structure. You have automatic calibration option as well as a manual option. This will give you better consistency with your prints. You have different options for receiving vessels, from microplate to multi-well plate to petri dish. Again, the benefit here being that you have compatibility with various vessel formats. The controls, you have an LCD screen with a knob or a software-mediated access that can be interfaced with a laptop on either Windows or Mac OS operating systems. And then last here, the materials, you have flexibility to use bioinks or hydrogels with or without cells. That includes Corning's Matrigel Matrix and collagen, as well as alginate-based bioinks. So the benefit overall is that this supports temperature-sensitive and ambient temperature hydrogels. For the basic operations of the MatriBot, it will depend on whether you want to do a dispense of a hydrogel or a bioink, but you do have flexibility when you're considering the different parameters for your print. And again, this can do droplet-based dispensing all the way through to multiple layer bioprint. So this will depend on the choice of your receiving dish. So in the examples here on the right, again, you've got the droplet delivery or a petri dish format where you're able to create much more complex structure. You have your choice of hydrogel or bioink, right? And even the viscosity, right? So whether you need low or high viscosity of that hydrogel, for example, different protein concentrations of Matrigel can be dispensed accurately. Again, you'll just have to customize the parameters to control things such as dispense speed, extrusion rate, and even retract volume. For example, size of objects can also be varied. So if you're doing droplet dispense, if you're just doing single droplet, right, greater than 3 microliters, so the example you're seeing here in the microplate, or you can do multiple droplet arrays in a single well. This slide focuses more specifically on the Matrigel droplet dispensing options. Again, a very commonly used workflow for bioprinting organoid culture. You're able to do single droplets on the lower left-hand set of images here. This is an example of a 3 microliter size droplet. And if you look in the middle image, you go up to 40 microliter size droplets. Again, flexibility depending on what your goal is, to whether it's a single dome or even multiple domes, which you're seeing here on the right-hand side with our 5 microliter per droplet, 4 droplets per well in our droplet array. An example application of using that droplet array print feature is in this application note, where we focused on using the Corning MatriBot bioprinter for high throughput 3D cell culture with our Corning Matrigel matrix. In this particular example, we use Matrigel at 9 mg per mil in a 24 well plate to dispense a 3 droplet array using 10 microliter droplet size volumes. Here we dispense 3 different types of cells. In the upper right hand panel, you're looking at the A549 cell dispensing. This is over a 10-day period. Same with the MDCK cells in the middle panel. So this is again growth over that 10-day period. And we also used a print for mouse intestinal organoids in the lower right-hand side set of images. So this was, of course, taken out over 7 days, just to give you an example of what these droplet arrays would look like if you were dispensing with cell embedded into the matrix. For the rest of the talk, I want to dive more deeply into the specific applications that we've developed at Corning using the different tools and technologies that I've talked about up to this point. The first application I want to talk to you about is an iPSC-derived intestinal organoid model for high-throughput screening. I'll first describe the intestinal organoid differentiation workflow that we use for this particular experiment, where we combined both a scaffold-free initial approach with a scaffolded approach while incorporating Corning Matrigel matrix for further differentiation of the intestinal organoid after day 10. So briefly, at day 0, we harvested our iPSC cells. This was a single cell suspension that was seeded into the spheroid microplate with 10 micromolar of the ROCK inhibitor. On day 1, we switched to definitive endoderm media for an additional 5 days. On day 6, we switched to the mid-hindgut media for 4 days. And then on day 10, this is where we overlaid with Corning Matrigel matrix, and we switched to our intestinal differentiation media for 4 weeks. I'll walk you through each stage of this differentiation protocol. This was the definitive endoderm stage, and this was after the spheroids were in culture, again in a scaffold-free spheroid microplate. This is after 6 days in culture. If we look just first at the bright field image, you can see a spherical formation of this intestinal organoid as we're starting. The markers here to confirm that we have definitive endoderm are SOX17 and CXCR4. So we saw immunofluorescence images that confirmed the presence of those 2 markers for definitive endoderm. Now we wanted to go beyond just what we're seeing with the immunofluorescence. We dissociated representative organoids at this stage and did flow cytometry analysis looking at both the SOX17 and the CXCR4 markers, which were both over 90% positive. So again, this gives us really positive indication that we do have definitive endoderm at this stage. The next stage of differentiation was assessment for mid-hindgut formation. After 10 days of culturing these spheroids in the spheroid microplate, if we look just first at bright field imaging, you can see a difference compared to the definitive endoderm. Now we have morphological change as the mid-hindgut is forming. This is positive via immunofluorescence for the CDX2 marker. So again, we saw really nice imaging there. But we wanted to be sure, just like we did at the definitive endoderm stage, so we dissociated into single cells that organoid and then looked via flow cytometry for CDX2 positivity. We saw over 80% positivity for the marker for CDX2, again confirming that these cells representative in that organoid were positive for mid-hindgut. After confirming that we had mid-hindgut formation, the next stage was to go towards full differentiation until a fully formed and defined intestinal organoid. This was after the full 4 weeks in culture, and keep in mind that after that day 10, we did have to add the Matrigel matrix to continue to drive differentiation and formation of this intestinal organoid. You have images here of the H&E staining showing that we had lumen formation with this culture. We also wanted to confirm functionality, so we did Schiff Alcyon blue staining to confirm mucus production with our intestinal organoid. In addition to the morphological changes from the H&E and the mucus production, we wanted to do a more thorough analysis that the organoid contained multiple intestinal cell lineages after the full 4 weeks in culture. Using immunofluorescence, we confirmed that we had goblet cells, mesenchymal cells, enterocytes, enteroendocrine cells, and Paneth cells all present with a fully defined and differentiated intestinal organoid. By combining a scaffold-free technology such as the spheroid microplate with a scaffold-based ECM such as Matrigel, we were able to combine and generate a reproducible high-throughput model system for studying the development of iPSC-derived intestinal organoids. This image shows a 96-well spheroid microplate containing intestinal organoids in each well. It's important to note that without the addition of Matrigel on day 10 in this protocol, further development of the intestinal organoid was not possible, confirming the critical role of the ECM for specific applications involving developmental modeling. As we move into our final application I want to discuss with you today, I wanted to briefly introduce the permeable supports that traditionally have been utilized for 2D cell culture and talk to them about how they're being repurposed for applications in 3D cell culture. Corning's permeable supports provide a dual compartment scaffold-based system with a microporous membrane bottom that can be combined with ECMs or substrates to create complex 3D matrix. These permeable supports come in different well sizes, membrane materials, and porosity, and they're compatible with a wide range of cell types. Now, traditionally, they've been involved in a 2D cell culture workflow supporting assays such as chemotaxis, invasion migration, co-culture, transport, and permeability. I'm going to talk to you about how they're being repurposed for applications with advanced 3D cell culture. This application, developed by our scientists at Corning, combines a spheroid microplate with a conical round well bottom with our 96 high throughput permeable support that plugs directly into that spheroid microplate. These can be combined for this application where we developed a 3D glioma blood-brain barrier model for high throughput testing of tumor cytotoxicity. There were different phases to generate this 3D cell culture model. The first stage was to establish an in vitro blood-brain barrier. To do this, we relied on a MDCK2 cell line that overexpresses MDR1. These cells were seeded at 100,000 cells per square centimeter. They were cultured for 5 days with a full media exchange on day 4 before assessing the effectiveness of that blood-brain barrier model on day 5. The schematic on the left shows you the concept that initially in our permeable support, we wanted to put this layer of MDCK2 cells and assess its ability to act as an in vitro blood-brain barrier. To establish our in vitro blood-brain barrier model, we used 2 different assays, the Lucifer Yellow assay and a Rhodamine 123 efflux assay. Starting first with the Lucifer Yellow permeability assay, this assay is to determine the effectiveness of having a non-leaky blood-brain barrier. By looking at Lucifer Yellow transport from the apical to the basolateral chamber, we can determine if this Lucifer Yellow compound can transport through the MDCK2 cell line barrier. Now, anything under 10 nanometer per second indicates that we do not have a leaky barrier, and so this was very well done at 2 nanometer per second, indicating that we had a tight formation of that monolayer in this basolateral chamber. The next stage was to look at efflux. So in a model of efflux, we want to use this Rhodamine 123 to determine if we have either apical to basolateral or basolateral to apical movement. And we anticipate that if we have a functional PGP pump, we should have basolateral to apical movement and not the reciprocal. And that's exactly what we saw here in this Rhodamine 123 flux assay, is that when we completed this, we saw basolateral to apical movement, and again, not the reciprocal. So with these two assays completed, we confirmed that we do have a functional model of our in vitro blood-brain barrier on our permeable support. In the next phase of this experimental design, we wanted to generate a 3D cell culture model for the gliomosphere assay using our spheroid microplate. To establish our glioblastoma spheroid, again, we used the LN229 glioblastoma cell lines. We seeded those into our 96-well spheroid microplate at 1,000 cells per well for 24 hours before removing the media and replacing with either a drug or negative control. We cultured for an additional 48 hours before using Promega Cell Titer Glow 3D reagent to assess cell viability via ATP. This is a representative image here in this middle showing the LN229 spheroid after 24 hours. And on the right, we were looking at LN229 cytotoxicity with cisplatin and piperlongumab drug compounds administered. Now, cisplatin we chose because we know this will not cross the blood-brain barrier. Piperlongumab was our compound candidate for the ability to cross the blood-brain barrier. So here, before we put it into the fully functional model with the blood-brain barrier intact, we wanted to assess viability, and we saw a good dose response curve for both of these drugs. Now that we have both our blood-brain barrier model and our 3D cell culture glioblastoma model, in this final stage, we can bring all of these components together. So in this oversimplification in this illustration, we're showing that we first validated our model for the blood-brain barrier. So again, this was the MDCK2 cells that overexpress MDR1. And also our model for the glioblastoma 3D cell culture model here with our spheroid microplate. Now again, on the left, I'll remind you that this oversimplification is not just a single transwell permeable support, but it is this high throughput screening 96 insert that fits directly into our 96-well spheroid microplate. I'll briefly describe the workflow to generate this 3D glioma model of the blood-brain barrier. On day 0, again, we seed the MDCK2 cells into our 96-well high throughput screening transwell. On day 4, we seed the LN229 cells into the spheroid microplate. On day 5, we then combine that transwell and the spheroid microplate and expose the apical chamber to drugs for 2 hours. After incubating with the drug, we remove that transwell, test that for monolayer integrity. We culture the spheroids for 2 additional days before performing an assay for cytotoxicity on day 7. In the final analysis from our 3D glioblastoma cell viability assay, we saw that administration of cisplatin in the absence of the blood-brain barrier was able to reduce the overall viability of those cells in 3D cell culture. Now, with the blood-brain barrier intact, the administration of cisplatin did not have a reduction in viability, confirming that indeed the blood-brain barrier could not be crossed by the cisplatin drug. Now, in the case of piperlongumab, without the blood-brain barrier, we did see a reduction in overall viability that was very similar to the conditions of the piperlongumab treatment with the blood-brain barrier. Now, the difference here is even with the blood-brain barrier intact, piperlongumab was able to cross that blood-brain barrier, whereas cisplatin was not. In summary, this model for the 3D glioblastoma cell viability assay does allow us to assess the ability for drugs to cross the blood-brain barrier as potential candidates for treatment and clinical application. To summarize what we talked about today, there were several key areas that we focused on when discussing 3D cell culture applications. We started off by focusing on ECMs, or a scaffold-based approach, then transitioned to a discussion on a scaffold-free approach, before digging deeper into the applications that focused on not only a single approach, but also the ability to combine both a scaffold and scaffold-free workflow when using these models for 3D cell culture. First, with the ECMs, I spoke about the Matrigel and how it supports a broad range of spheroid and organoid assays and applications, and how that Matrigel can be combined with other ECMs such as collagen for certain assays and applications. I illustrated the example for breast cancer organoids, where combining collagen and Matrigel was used to accomplish an invasion assay, versus the assays that can be completed with a pure Matrigel or a pure collagen, again, talking about the importance of that ECM choice to accomplish your desired endpoint assay. Then moved into a discussion on a scaffold and scaffold-free assay-ready options. Those include the spheroid and Alplasia microplates that support that scaffold-free generation of spheroids and organoids with the same ultra-low attachment surface option. Whereas the Matrigel Matrix 3D plates, they provide that same assay-ready solution for those applications that require a scaffold-based approach, such as our airway organoids. I then talked about the MatriBot bioprinter as one of our new products that provides a more automated option to incorporate dispensing or printing workflows for assays that incorporate the use of ECMs or bioinks. In our final application section, I talked about a scaffold-free disease modeling with our tumor spheroids, a scaffold-based disease modeling approach with our airway organoids, a combined approach for developmental modeling of our intestinal organoids, and then I talked about an example from the literature where a combined approach of both a scaffold-free and a scaffolded approach was utilized for dynamic conditions for developmental modeling with brain organoids. Now, depending on your application, your 3D cell culture experiment may require a single approach using a scaffold or scaffold-free option, or a combination of both of these approaches. Thank you so much for joining us today to learn about Corning's portfolio of 3D cell culture products. We're excited about the new products that we're able to offer with our Matrigel Matrix 3D plates and our Corning MatriBot bioprinter. Happy to address any questions about our products and the application notes that I shared to help you advance your research in the 3D cell culture space.